Alright, welcome to the second part of our looking at proteins. Um, in the first part, we looked at amino acids, which are the building blocks of proteins, and we looked at how we connect amino acids together through condensation reactions to create polypeptide bonds, and those are the beginning formations of our protein. Now what we're going to do is look at the different structures of proteins and the characteristics of proteins when they're folded. What you see here in front of you is a computer-generated diagram of a protein that has been completely folded. Now, when a protein folds into its correct shape, three-dimensional shape, it will determine the actual function of the protein. So it is very important that a protein folds properly in order to create a functional protein. And different proteins will have different three-dimensional shapes, which results in different functions in cells. And so what we're going to do is look at these different structures of proteins and how proteins are folded into different shapes and go from there. All right. So let's go to our next slide here. Here are the four main levels of protein structure: primary, secondary, tertiary, and quaternary. You need to learn these four levels of protein structure very well for the IB exam. We are going to look at the details of each of these, starting with primary structure. This graph table more or less shows you the different levels of structure of a protein. The first one up there you see is primary structure. That is the actual amino acid sequence in the correct order. And then that amino acid chain is, or polypeptide chain we should say, is going to start folding into secondary structures, and then they will fold into the final protein structure, which is called the tertiary structure. And then sometimes the tertiary structures of one protein will form a bond between a tertiary structure of another protein, and that will end up creating a quaternary structure, or a gigantic protein. And so we'll look at each one of these levels in more detail. Alright, so the first one we're going to look at is primary structure, or the precise sequence of amino acids. Well, how is the precise sequence of amino acids determined? Well, it is determined by your DNA, your genetic makeup. So the DNA in your cells contains all the information necessary to make a protein. This is called a gene. And DNA gene codes or makes another molecule called messenger RNA. And just as it sounds, messenger RNA carries the message to make a protein. And that messenger RNA has the correct message to put the right amino acids in the correct sequence to make our polypeptide chain. And all we do is basically take one amino acid and put it in the right order, and through a condensation reaction we add the next amino acid that we need to add, such as you can see here. Our first amino acid is lysine, and then we have valine, and then so forth, and we just keep on creating our chain all the way till we get to the very end of our polypeptide sequence. Alright, so let's erase this. Okay, now as we stated in our last lecture, we have the N terminus and the C terminus. The N terminal end is over here, and over here at the very end of our protein that we're making is the C terminus. Okay, that will be our C terminus, right? Now, what we're going to do is we're going to erase this, and we're going to make a protein sequence here. And our first one, we're going to start out with cysteine. And we're going to add our next amino acid, arginine. Now, this line represents here the peptide bond that exists between these two amino acids. And notice I'm using the three-letter abbreviations. And you can get the three-letter abbreviations in your IB data packlet that we went over last time. And remember that we're always going to add the next amino acid to the C terminal end. Always to the C terminal end. So there's valine. And then again at the C terminal end we'll add our next amino acid. And again, the sequence, the order of each one of these amino acids is determined by our genetic makeup. And that sequence needs to be precise, needs to be very good. Now, if another amino acid is incorporated in there in the wrong place, that can result in the wrong folding of a protein. So really, the precise amino acid sequence is going to help to determine the overall three-dimensional structure or shape of the protein. So the primary structure is very important to get it right. And if we don't get it right, the protein may fold wrong and result in a protein that does not do what it's supposed to do, the wrong function. And if it doesn't do the right function, it could lead to serious consequences to our human bodies, even death. And so when a mutation happens in a gene, which means that our message changes, it could create the wrong amino acid sequence, where therefore results in creating the wrong protein or the wrong protein shape, so it cannot carry out the correct function. So I hope you understand the importance of the primary structure and that we need to get it right. The other thing you also have to remember is we always add amino acids to the C terminal end right here. And we can add our next one, such as histine and so forth. Alright, there's primary structure. Okay, next comes, once the amino acid sequence, the correct one, is put together, and we have our whole primary structure now, our protein begins to make its first folds. And the first folds are probably with secondary structures. They don't have to be, but probably in many cases secondary structures begin to come first. Now, secondary structures consist of regular repeated patterns in different regions in the polypeptide chain. Now, this is mainly influenced by hydrogen bonds that occur basically between, went ahead, so let me go back, between the peptide come down here, peptide bonds of one amino acid and another. Okay, so again, we're going to form hydrogen bonds between one amino acid, peptide bond of one amino acid, and the peptide bond of another amino acid. So erase that here. And so you can kind of look at it like this. You have a C, there's part of the peptide bond of one amino acid group, and it's going to form a hydrogen bond, between a peptide bond of another amino acid. Okay? And that hydrogen bond exists between the oxygen in the double carbon bond and the hydrogen that's located on the amine group. So, all right. So we're going to look at the two important secondary structures, alpha helices and beta plated sheets now. All right. We're going to look at alpha helices first. Now, alpha helix are very common secondary structures in proteins. You need to know some common characteristics about alpha helices and some examples of proteins that contain alpha helices, which are listed here on this slide. So first of all, what happens is we form a hydrogen bond. Oh, we went ahead again. Okay, here we go. So we're going to form a hydrogen bond between the carbonyl oxygen and the amino acid, like we saw before, in the peptide bond itself. And when we create alpha helices, there is a twist in the chain as a result of forming these hydrogen bonds between the carbonyl oxygen and the amino hydrogen, basically, in the polypeptide chain. This twist results in a right-hand coil or an alpha helix. Now, some common ideas here is that there is 3.6 amino acids per turn, which you need to know, which is labeled right here. So every turn in the helical structure, every turn that we have from here to here is 3.6 amino acids. And each of them are forming these hydrogen bonds. You can see here a hydrogen bond between the peptide bond of one amino acid and the peptide bond of another amino acid. Now, some common proteins that contain helices are like keratins, which are found in hair. Feathers, hooves. Keratin is a very common protein. Collagen, which is also found in hair, are all made up of these alpha helices. They're flexible and elastic. Okay, so alpha helices are both flexible and elastic, which makes them great proteins, or help to make great proteins that need that flexibility and elasticity, such as in hair and feathers. Alright, so now what we're going to do is look at beta plated sheets. Now, they're a little bit different. They still are forming hydrogen bonds between the NH group okay, between the NH group and the CO group on different amino acids. But they're going to be, instead of a helical structure, they're going to create parallel regions to each other, which we'll show you a diagram here in a minute. These beta-plated sheets, they are flexible. They're flexible, but they are inelastic. So they don't stretch very well. So where we find those is like in spider webs, silk. When you look at a spider web, they're usually composed of lots of beta-plated sheets where they're flexible, but they're not elastic, so they don't stretch very well. All right. Alright, so here is example of beta-plated sheets. And you can see that instead of being helical structure, you have more or less some parallel. So here's our primary chain is going in this direction, and then there's going to be a turn. And this turn is called a hairpin turn, or a hairpin loop. And it's going. There usually is 2-3 amino acids that are involved in that turn, and then our sequence picks up again at the turn. Now that's what the results of this is creating two parallel strands in our primary structure. And when those two strands that are parallel, they're going to begin to form hydrogen bonds between each other, as you can see here. And those hydrogen bonds are going to connect both of those strands together to make these beta-plated sheets. Here is a closer look at beta-plated sheets. We can see here, this is the carbon-hydrogen double bond, and this is the nitrogen-hydrogen in another polypeptide bond in another amino acid, and we have the hydrogen bond between them. Same thing here. Here's the peptide bond area, peptide bond of another, and we form these hydrogen bonds. And that creates a very strong, rigid structure, but a fold nevertheless in the protein. Alright, now, after we begin to fold our protein into secondary structures, then we begin to make our final protein folds into our three-dimensional structure. The final folds of the protein is in the tertiary structure. Now, this final three-dimensional shape of the protein results in the amino acid R groups, okay, or interactions between the R groups. So with secondary structures, it was interactions between the peptide bonds of different amino acids. And now for the tertiary structure, or for the final folds that are made, there are interactions between the R groups of the amino acids. Now, if you remember, that, oops, I will go back to this. Okay, if you remember that the R groups have different characteristics. We have nonpolar R groups, like in glycine here, and alanine, and valine. These are all nonpolar, so they do not carry a charge at all. And these nonpolar amino acid side chains, or R groups, are going to interact with each other. And these interactions And these interactions form what are called hydrophobic interactions. Okay, and we'll talk more about them a little bit later. We also have polar side groups, like in serine and thorine and cysteine. Those are all polar groups, and they're going to have charges or partial charges. And then we have our acid groups and our base groups that are also going to have charges, and all of these are going to interact to create, help to create our tertiary structure of our protein. Okay, so we go back to this slide here, and this slide is showing more or less some possible interactions between our groups. So we have hydrophobic interactions that you need to become aware of. We have disulfide bridges or disulfide bonds. We have ionic bonding that occurs, and we have hydrogen bonds. All four of these interactions between our groups you need to know, and know well, very well. Okay? So the first ones that we're going to look at are non-covalent bonds. Okay? And the first non-covalent bonds are the ones that are formed hydrogen bonding. Okay? Hydrogen bonding here. That's what we're going to look at. And those are going to be between polar side chains, or polar R groups. Okay, so hydrogen bonding takes place between R groups that are polar. And then what we get is elastic interactions. Elastic interactions are also ionic bonding. So these are like ionic bonds. Okay, so elastic interactions, another word for them, are ionic bonds, and those take place between basically R groups that have a charge. Okay. So again, hydrogen bonding is between R groups that are polar, and electrostatic interactions or ionic bonds are between R groups that carry a charge. Okay, we'll erase this for a minute. And our last group that are non-covalent bonded are hydrophobic interactions, which we kind of hinted at. And these are really literally between non-R groups, non-polar R groups. All non-polar R groups create hydrophobic interactions. Or in other words, these are weak van der Waals forces. Okay, which you're very familiar with van der Waals forces. Where we find weak van der Waals forces or these hydrophobic interactions in proteins is in the nonpolar regions of the molecule, or more or less the interior of the protein. So the interior of the protein contains hydrophobic interactions That is important to know. Where you find hydrogen bonding and ionic bonds are towards the outside of the protein, which is directly in contact with water. But the hydrophobic, meaning that it does not like water, phobic meaning it hates water. Those kind of interactions are found in the center or the interior of the protein. And these hydrophobic interactions are going to strengthen the protein in the interior. Okay, so now some non-covalent bonds that you need to be familiar with. The most important one is the disulfide bond between two cysteine residues or two cysteine amino acids. Basically, it would look very much something like you would have a CH2 and then the S on the cysteine, and then you have another cysteine residue very close, and they're going to form a covalent bond, or what we call a disulfide bond, between two cysteine residues, and that's between the two sulfurs on cysteine. So look at your data packet, look at cysteine, and you'll see that it has a CH group on there. We lose the H, or the SH, we lose the H, and we form a disulfide bond between the two sulfur groups in two different cysteine amino acids. Okay, and those really help to stabilize the structure. They are also found to help to make quaternary structures. So we take different tertiary structures. Once the protein is folded, we have the final tertiary structure, and through disulfide bonds we can bond another tertiary structure together to create a gigantic protein. Okay, so here's a diagram of all the different all the different basically interactions between the R groups that can be made. We'll point these out. You have hydrophobic interactions between nonpolar groups. We have electrostatic attractions between charged. Notice they're charged here. So these are ionic bonds between these charged side chains. We have hydrogen bonding that occurs between polar side chains. Again, hydrogen bonds between polar side chains, ionic bonding between charged side chains, and hydrophobic interactions between non-polar side chains. You need to understand that. And then we have disulfide bonds between two cysteines. And all of these interactions create a folded protein. And once that protein is folded into its secondary and tertiary structures, then that protein is capable of doing and completing its proper function. So make sure you read through this in your book, and if you have specific questions, let me know. But you really need to know these interactions. Again, ionic interactions, hydrophobic interactions, hydrogen bond interactions, and disulfide bonds.